And at this point, I welcome everybody to our third webinar, the third International Legume Society webinar. I'm Nelson Nazzicari from CREA, Italy, and today we are going to listen from Julia Buitink. I hope to have pronounced the name correctly, about improvement of seed vigor, or more formally, physiological seed quality of legumes acquisition and expression. This is our third webinar. We started with 2 months ago with Christian Wick, with a more general discussion about the opportunities and what does it mean to breed for legumes in Europe and what are the consequences. Last month we went a little bit more on the details with Peter Smikal, and we talked about domestication and the effect of domestication on the expected and unexpected effects of domestication in legumes. Both these webinars have been recorded and are available on our website at the International Legume Society website. This is our third installment, and today we have the pleasure to have Julia Buitink, which is, among other things, the president of the International Society for Seed Science. She's based in Angers, in France, and I've put her contact here and are in the website, on the Twitter account, on ResearchGate, everywhere. I've tried to make her as public as possible. So, without further ado, I thank Julia for her time, and I will stop sharing, and will pass her the microphone. As usual, if you have questions, I will monitor the chat, so we can discuss if we have any burning question. We can decide. Julia can tell us if she prefers to be interrupted if there are questions, or usually we have time after the webinar, and there is time for a few questions. But it's up to you to accept it if somebody raises their hand. So thanks again. I remind everybody that the webinar is being recorded, so if you talk, or if your camera is on, you will be recorded and you will end up on our YouTube channel. Please be aware of this. And please, Julia, welcome, and thanks for your presentation.
Okay, well, thank you very much for this introduction. I'm just going to open my presentation again because I just closed it. Up. Okay, do you see my presentation?
Yes.
Yes. Okay. Perfect. Yeah, because I don't have any additional screen. Okay. Great. Well, first of all, I would like to thank the International Legume Society and Paolo and Nelson for inviting me to present some of the issues related to seed quality, and especially legume seed quality. And so the title that I gave it is Physiological Seed Quality, which is what seed vigor is about, and I will explain about that. Of legumes, the acquisition and expression. And so just to introduce quickly the research team in which I'm working. So we're at the Research Institute of Horticulture and Seeds, which is in Angers. That was just said. This is the map of France, and you can see Angers here. So we're in the Loire region. This is, of course, not our laboratory. This is the beautiful castle in Angers that's of the 13th century. And this is, in fact, our lab here. Do you see my pointer? Probably not. Just a moment. Okay, this is our lab here, and is our big greenhouse installation, and so this is where we are. And within this research institute, one of the 12 teams is the seed team, and that's what I'm working in together with Olivier Leprince and Jérôme Verdier, which some of you know as well, I think, and together with our technical staff that help us with all the experiments. So this is the layout of the presentation. I expect it to last about 35, 40 minutes, I think. We'll see how fast it goes. So we'll talk about the definition of seed vigor, the acquisition, when is it acquired? The markers of the maturation program, how do we know how far we are with this acquisition? I will talk a bit about seed vigor and its proxy for longevity, the storability, and the issues that we have, especially related to legumes. I will talk about the mechanisms that confer longevity, so more like the molecular mechanisms. So how do seeds survive this drying and retain the seed vigor? And then I will have just a couple of slides on the effect of environment on seed vigor traits. We'll talk about genotypes times environment interaction and phenotypic plasticity.
I'm sorry. Please, Olivier, could you mute your? Oh, I'm going to mute you. I'm going to mute everybody because I'm hearing some noise coming from the guest. I'm sorry, Julia. Please go on.
That's okay. Okay, so let me continue first. So physiological seed quality, as I said, that's the term that we use. We also call it seed vigor, and in fact, what we know is that crop yield and resource use efficiency depend on successful plant establishment in the field. And it's especially the vigor of the seeds that defines their ability to germinate and establish seedlings. That is defined as their ability to germinate and establish seedlings rapidly, uniformly, and robustly across diverse environmental conditions. And a key component of the performance of crop seed is seed vigor, which is really a complex trait. So here I just show you a field of seeds that were sown at different seed quality, different physiological seed quality, and you really see everything is emerged, but in terms of the delay of emergence or the homogeneity of emergence, we see big differences. Okay. So if we look at the definition of seed vigor, I took back the official book of the International Seed Testing Association. And it says that seed vigor is the sum of those properties that determine the activity and performance of seed lots of acceptable germination in a wide range of environments. So as you can see from the definition, it's not a single measurable property, but it's a concept that is associated with aspects of seed performance. And if we see what kind of aspects this includes, it is the rate of seed germination, so the final percentage of germination. That is, of course, important for stand establishment. It's the uniformity of the seed germination. Will it all germinate at the same time, or will it be a long difference between the first seed and the last seed in your seed batch? It's also the rate and uniformity of seedling growth. And it's the emergence ability of the seeds under unfavorable environmental conditions, of course, with biotic and abiotic stress being the main factors. Next to these vigor measurements, there's another type of vigor that we talk about, which is the performance after storage. And that's particularly the retention of this seed vigor. So if we store seeds, the longer we store them, the more damage will occur during storage, and then the more seed vigor will be decreased, and I will show this in my presentation a little bit later on. So as a final conclusion, we can talk about vigor as a potential performance of viable seeds in agricultural practice, and this is determined by the complex interaction between the genetic and the environment components. Okay, well, I started to directly talk, I wanted to directly talk about the issues that mainly legume seeds have, and this is also because some of the legume seeds are quite big. If we see where the problems are in terms of having successful crops for legumes, we know it begins with obtaining rapid emergence and adequate stands. And this is really, if things go wrong, where we can see that they go wrong. So there are two things that, there are two issues. One is traced back to the seed quality, so the initial seed quality of the seed batch that's going to be sown. So that means seeds that have acquired, yes or no, correctly their seed vigor. That is what I will be talking about in the first part. And then what happened with the seed in the time between seed planting and emergence. So what they experience in the field. Now, if we look at these special issues in relation to legume seeds, we talk about slow emergence, especially in the cold. And then I'm talking, for instance, about pea, I'm talking about soybean, I'm talking about faba bean. And this means is because they emerge very slowly, this gives competition with weeds. This gives problem of damping off, so interaction with the pathogens that are in the soil, and at the end will affect will affect the stand establishment. A second issue that especially bean and soybean seeds have is imbibitional damage. Imbibitional damage is related to the speed at which the seeds imbibe, and this is related to the seed coat properties. And this is exacerbated in the cold, and that means if you do early sowing, for instance, you have to watch out that the temperature of the soil is high enough not to have this imbibitional problem. Third of all, we know there's a strong environmental effect on the seed quality acquisition. I'll show you some of the results, and this gives, of course, then very unreproducible results in terms of yield, depending on the environment. And this is, of course, getting more and more important when we think about climate change. And then more specific issues that are not only related to legumes, but for instance, play a big role in seeds like soybean. You can have production of green seeds due to drought or heat during the seed production, so where the maturation is truncated, and this affects also the nutritional quality, but also the physiological seed quality. Bad storability, especially in terms of soybeans, so this gives you problems with seed vigor after storing the seed lots, or if you want to carry over the seed lots to the next year. And then there's this problem of legume seeds, of course, with the presence of anti-nutritional components. You say, well, why do you talk about it? Well, I'll come back to some of them later, especially in relation to oligosaccharides, because if you want to remove them, we have to make sure that this doesn't affect anything that is important in terms of seed vigor. So what I show you here is a scheme where you have the different phases of going from a seed, a dry seed, completely on your left, to a small seedling. So first we have the imbibition phase. As I said, cold temperatures or having dried the seeds to very low moisture content before imbibition can actually give this imbibitional injury. So this is the first important step. Then after the imbibition, what you will have is at a certain point the germination. And then of course the speed of germination is important, but also the final percentage of seeds that will germinate. And then after germination, still going on in the soil here is the heterotrophic phase. And this is where you have the radicle that will grow, that will actually go down, it will search for water. And at a certain point, you have your, in the case of dicotyledons, your hypocotyl going out here, or the epicotyl when you talk about pea. Then there is the perception of light, and then we go into autotrophic growth, where actually we'll have chlorophyll that will be produced in the cotyledons. The cotyledons will open. This is, of course, in a dicot, like soybean, for instance, or alfalfa, or red clover. And at a certain point, you'll have the true leaves that will be appearing. Environment will play an important role throughout the different phases. And you can see already here, if we talk about seed vigor, it's not really one thing, it's not really one phase. It can be related to imbibitional problems, having hard-seeded issues. That's what Pietro talked about last webinar. I won't go into that. You can have problems down in the soil in terms of pathogens or having too cold, and then you can have problems here in terms of having stunted growth, for instance. So when is this seed vigor, or when are these different seed vigor traits acquired? And that's what I would like to show you here, based on a couple of slides from a model system that we have been working on a lot in our laboratory. It's Medicaca truncatula, baromatic. And what you can see here is a scheme going from flowering, from pollination, seed filling here, until what we call late maturation, where you have the loss of chlorophyll onto a dry seed. Here you see the same results, but in numbers, so days after pollination on the x-axis. And here, first of all, what you can see is the dry weight. And the dry weight here in green, you can see it increases until about 32 days after pollination. These are under conditions of 20 degrees during the day and 18 degrees at night in our greenhouse. And you can see that after that dry weight has accumulated, there's no change anymore. So seed filling stops at about 28, 30 days after pollination under our conditions. And at the same time, when we look at the water content, it actually goes down simply because the cells are getting filled, so the water is being pushed out. In terms of reserve accumulation, Medicago truncatula is a proteinous seed, so you have mainly the accumulation of legumes and physalins. This is data from Karin Gallardo. You can see that's occurring here. And then a little bit later during maturation, you have the accumulation of sugars. And that's what you can see here in red. This is stachyose. It's a tetrasaccharide. So it's a sucrose molecule, a disaccharide, in which we add two galactose molecules to have a tetrasaccharide. A trisaccharide is raffinose and a sucrose is a disaccharide. And if you look what happens with sucrose, it's actually going down because, as you can see, it is used to form your stachyose. But as you can see as well, this stachyose is formed later, there where seed filling is ending. So what goes on then in terms of all these seed figure traits? Let's first see what happens with percentage of germination. When you take seeds here at 16 days after pollination, you take them out of the pots and you put them in a petri dish with some water. They do not germinate. They have not yet acquired the capacity to germinate. A couple of days later, we see that it has. So here at 20 days or 21 days after pollination, you put these seeds in a petri dish and you see the radicle protruding through the seed coat, which is the germination, and they grow into little seedlings. At a certain point here, what you can see is you have the germination actually going down. This is of course not because the seeds lose their viability, but this is because they go into dormancy, and then there's hard-seeded dormancy, but that is not this here. What you can see, we scarified the seeds. This is physical dormancy. And this is kind of a wild species trait. If we don't look now at only if they germinate, yes or no, but we look at how long they take to germinate, so the speed of germination, and often we quantified it by the time it takes for the whole seed population to germinate for 50%, you can see that the further you go with the maturation, the faster the seeds will germinate. So this is the first aspect or the second aspect of seed vigor. So first aspect is the percentage of germination, and the second aspect is the speed of germination. And what you can see, this speed starts to be optimal here when the chlorophyll starts to be lost. So that is germination. If we now take these seeds here at 21 days after pollination, so they germinate very happily, and you dry them back rapidly, and you re-imbibe them, they're dead. However, there is this very special capacity that these seeds acquire during this maturation, which you see in the red line here, which is the acquisition of desiccation tolerance. If we take seeds out of their pots at 24 days, 26 days after pollination here, we dry them rapidly, we rehydrate them, you can see they perfectly are capable of surviving this drying. Now, we have been working quite a lot on this desiccation tolerance. I will just show you 2 slides. First, to make you understand how dry is dry. Yeah? So the desiccation tolerance, the definition is the ability to survive the removal of all or almost all of the cellular water without irreversible damage. So 3 measures of dryness you can see here: the water potential, the osmolarity, or the relative humidity. And here you see the biological response. And when we dry out our vegetative material, for instance, plants, we can see they start to wilt, but depending on the type of plants we are looking at between -2 and -5 megapascals. A little bit further goes into lethal wilting. If we grow further, everything that is an anhydrobiote, meaning a tissue that is desiccation tolerant, such as most pollens, most seeds, you will actually stop the growth. And in dry seeds or plants, which are resurrection plants, which can survive complete drying, you have a water potential in the cytoplasm that's around -200 megapascals. So this is really, really dry. Now, the fact that these seeds are dry gives them exceptional biological properties. And in fact, what it makes them extremely tolerant to different conditions of the environment. You can put them under complete anoxia If you rehydrate them, they're still alive. Radiations then can support quite a lot of radiations. You can put them in liquid nitrogen. You can throw them in liquid nitrogen. They will be perfectly happy. You can heat them up for a short time to 150 degrees, not too long, because then if they're starchy, they might pop and you have popcorn. You can put them under vacuum and you can dry them to really, really, really low relative humidities. The second particular aspect of having dry seeds is that there's no true metabolism detectable in the dry state. And this, of course, also means that there's less deteriorative reactions that will be going on. Okay, so I've been talking to you about this acquisition of desiccation tolerance. But if we look here where seeds have acquired this capacity to be dry, if you keep them for a while on the bench, you store them, they lose their viability very, very fast. And that is what we talk about. And that is the definition of longevity. And how do we measure this longevity? So its definition is the time that the seed population can remain alive during storage. And the way we measure this, measure it, we take the seeds, we equilibrate them to a certain relative humidity at which we want to do our storage experiment. And this is, of course, often a little bit higher than the actual storage that we do in a gene bank or that we do under natural storage, because we want to have the results in a couple of months. And then when we have equilibrated these seeds at 60% relative humidity or 75% relative humidity, we either do open storage, so we put them in these boxes as you see here, so in presence of oxygen, or we close them in hermetically sealed bags, and that's what we call closed storage where there is no oxygen. And of course, storage will go, or longevity will be higher when you put them in closed storage when there's no oxygen than when there's oxygen. Okay, so we store these seeds at different time points during storage. So here you see the experiment. We did it at 75% relative humidity at 35 degrees. We took the seeds at different maturation stages. So here in brown, seeds at 28 days after pollination, and we store them. And the moment we start storing them, what you can see is that we decrease, the germination decreases, and actually the seeds die. The viability decreases. Now, the longer we wait in terms of letting maturation going on, the longer we can store the seeds before the viability drops. This is at abscission stage. Now, to quantify this P50, to quantify this P50 is that we take the time of storage for 50% of the population to have lost their viability. And when we calculate this P50 for all the different maturation stages, and we plot it here, you can see in the brown line, the longevity increases gradually during maturation, about a 30-fold. Now, of course, you can understand directly that if you harvest too early, your seeds won't have yet acquired their longevity. And this is important. Why? Because this longevity, except for having viable seeds after storage, also can be used as a proxy for seed vigor. And I'll go into that in a moment. So it's not only longevity that is acquired here at this final maturation stage, so there where chlorophyll starts to be lost, but also, for instance, here what I've plotted to you is salt stress tolerance. So we put the seeds in a solution, salt solution, and a cell solution, and we looked how long it took for them to germinate or whether they germinated. And you see mature seeds germinate for about 80%, but immature seeds have difficulty germinating. They haven't yet acquired their tolerance. So this is in Medicago truncatula. And then you can say, yeah, okay, well, fine. But what about the seed maturation program in other legumes? And another species that we work a lot on in the laboratory is soybean. And so here what you can see, and this is work that we published a couple of years ago, the developmental stages of the soybean seeds, going from stage 5, 6, 7, 8, and then 9, which is the final mature stage. And you can see a little bit the same thing. It's a bit busy graph, but here again, we have in green here the acquisition of germination. We have in red the acquisition of desiccation tolerance, and we have in brown the acquisition of longevity. And you might notice here the stage 7 of soybean, we actually took a lot of substages because we realized that this is really where the longevity and the desiccation tolerance is being acquired. Okay, just to stay a bit on soybeans. So as you showed for Medicaca truncatula, you had these oligosaccharides, this raffinose family oligosaccharides that were increasing. So what's going on in soybean? This is a little bit the same. It's just a little bit more comprehensive. Here you see the sugar contents either in the axis of the seeds and in the cotyledons during, the axis is here, days after flowering, what you can see here. In gray is the acquisition of longevity. First of all, here you see the monosaccharides. As you can see, they really go down and are almost low when the seeds are dry. You see your sucrose that kind of stays high and goes down at the bottom. And you see the stachyose here. The raffinose doesn't go up much. It's very intermediary, but you see the stachyose increasing a little bit prior to the acquisition of longevity. And when you look at what happens in the cotyledons, the stachyose is actually being accumulated at the same time of longevity. So it's not only these sugars that change, it's not only the salt tolerance. Here is another measurements that we looked at. This is Denise Basso, a Brazilian PhD student that was in our lab for 1 year. She looked at the length of the hypocotyl after 7 days of incubation in the dark of the seeds. And there again, you can see that during this maturation, it's really acquiring from this stage 7.1.3 onwards, and its maximum at stage 9. Now, the other information that you can see here, and what I just would like to show you, germination is 100%. So we really talk about seedling characteristics that are also being acquired during this maturation. And here, just what I would like to show you is seeds that were harvested at 3 different places on the plant, on the top, from the middle, or the bottom. And there also you can see that depending where the seeds are being produced, this will play on a seed vigor trait, which will be the length of the hypocotyl that it can travel before going out of the soil. So if we compare both maturation programs, this is what you can see. So this is Medicaca truncatula, this is soybean here. As you can see, of course, in Medicaca truncatula, you have little oil, you have a lot of proteins. This is different in soybean, but what you can see is the same as the accumulation of soluble sugars, especially the oligosaccharides from the raffinose family oligosaccharides. And here the same thing. And then the acquisition of the survival in the dry state with first desiccation tolerance being acquired in a very short window, and then a gradual increase of the longevity, the storability of the seeds. And just to tell you that the main markers of seed longevity, you can see it directly, and of seed vigor are oligosaccharide increase and the loss of chlorophyll. How does this compare to our non-legume seeds? Well, it's about the same thing. I just want to show you here that in terms of oligosaccharides, this is really something that is accumulated a lot in legume seeds, much less in other species. You can see there's a little bit increase in raffinose, but that does coincide with the increase of the storability of the longevity of the seeds. So why do I talk about this longevity? Well, I already said it several times. Longevity is a key component of seed vigor, and it's a proxy for seed vigor. Why is that? Well, here I show you results of a recombinant inbred line population that we worked on, LR4 for Medicaca truncatula. This shows you the germination curves of real 160, so the percentage of germination during imbibition of either the control seeds, so you see they germinate between 15 and 25 hours, and of a seed lot that has been aged for 12 days under our accelerated aging conditions. And what you can see is not only the decrease in the percentage of seeds that germinate, but also a decrease in the time to reach 50% germination. And next to this speed of germination that is different, you also can see that the percentage of normal seedlings after aging is different. There's many more normal seedlings for one of the parents here than for the second parent. So we have the percentage of germination that decreases during aging, we have the speed of aging that decreases, and we have the normal seedlings that decrease. And in fact, if you now go to the field with all these results and you look at germination tests in the laboratory, germination tests, you can see it, for instance, 48 days after storage, the seed lot can still germinate for 100%. But if you sow these seeds in the field, you actually see that the seedling emergence has already decreased to 30%. So this shows us that we have to watch out not to look only at germination, but also at the speed of germination or the normality of the seedlings. The whole, everything I told you about is kind of summarized here. So here what you can see is during seed development. This is here, this part is seed development. The seeds are dry harvested here at these arrows, and then they're being stored. Germination is being acquired, so this is soybean seeds at a certain point during maturation. Normal seedlings, the percentage goes up a little bit later during maturation, and really the vigor is acquired even later. So that's already what I showed you. But what I want to point your attention to is here, this part of storage. If you store your different seed lots, so let's say we take 4 seed lots, A, B, C, and D, having stored for a longer amount of time. So if you store seed at a certain point, germination percentage will go down. But already earlier during storage, you'll have your normal seedlings that go down, and even earlier during storage, your vigor will go down. And that's what you can see here. Oops, here in these graphs. Time from sowing. So if you store the seeds longer, your seed curves will decay. And also this, if you now sow these seed lots under different stresses, increasing stresses, for instance, cold temperature, you can see that the longer the seed lot was stored, here, the seed lot D, the worse they perform under stress. One last thing, kind of an idea about how on storability, how long do seeds have to be stored in order to die? Well, of course, this is species dependent. Soybean is a notably bad store. Pea stores much longer, but we can say something about the external conditions in which we put the seeds in. In fact, there are 2 conditions. One is the relative humidity or the moisture content that is accumulated in the seeds, and the second one is the temperature. And what you can see here, this is the relationship between temperature for Medicago truncatula seeds. So this is the P50, so on the log scale, so it's the time that 50% of the seed lot dies. And you can see the lower the temperature, first of all, the higher the longevity, which is not surprising, the colder the better. We also know if you go too dry, below 11%, this is really detrimental. You remove the last water molecules around the shell. But there is this rule of thumb that is quite interesting, and it works for Medicago. It's Harrington's thumb rules that proposes that the time for 50% of the seeds to die doubles for every 5 degrees reduction in temperature or 1% reduction in moisture level. So if you wonder about your seed batch and you want to know how much more you want to make them stay alive, you can use this rule. So what is then happening during this late maturation? Well, there are 3 mechanisms that are being acquired during this development. One is protection, to avoid the oxidation of proteins, lipids, and nucleic acid, to prevent the structural changes in proteins and membrane phospholipids, and to prevent structural collapse. So you don't want to have big vacuoles, then when you dry them out, the whole cell collapses. There is damage control. We have to remove molecules or structures that can become damaging during drying. And in terms of repair, we have to restore the genome integrity and eliminate aggregated proteins and oxidized lipids and proteins. So I've got a couple of slides to show you the main mechanisms that the seeds put in place during this maturation. And the first one, and probably one of the most important ones, is the seed coat. And I don't have a picture here of legume seeds. I show you a picture because it's nicely colored in terms of what type of molecules are found where in seed coat of Arabidopsis of rice, but you can extrapolate this to legume species. You have the accumulation of flavonoid pigments, such as proanthocyanins in the endothelium and in the parenchymatic cells of the seed coat. They function to decrease the permeability against oxygen and have antioxidant properties. There are layers of lipid polyesters, such as cutin, and suberin. They seal also off the embryos from the outside oxygen. There are lignans that are being produced. They provide increased resistance to mechanical damage. We know this for soybean seeds, and they reduce the permeability, so the speed of imbibition. And there's activation of biotic defense genes that actually also help reinforcing the seed coat. Within the embryo, we have protection on 2 levels. First of all, protection against oxidation. And this is done by the synthesis of lipophilic antioxidants, such as lipocalins and tocopherol molecules, and the presence of hydrophilic antioxidants, such as reduced glutathione. So that is the oxidation, the protection against oxidation. And in terms of structural protection, there's a number of hydrophilic molecules that are being synthesized during this late maturation phase. And I'll show you 2 families that are the most important. So the first thing I already talked to you about are these non-reducing sugars, sucrose, or sugars of the raffinose oligosaccharide families, and late embryogenesis abundant proteins. Now, these are kind of interesting proteins. They're very hydrophilic. They dissolve very well in water. And we have shown that if you remove them, here you see 2 lines. If you remove them from seeds, so this is the longevity, the P50 of the wild type seeds. If you remove the hydrins, you can see that actually you reduce the longevity by 2-fold. So these molecules are really important to maintain the structural integrity. Now, how does this work? Both of these molecules actually work via a similar mechanism. And the mechanisms depends on the hydration state of the cells. So here you go, cells that are fully hydrated. So this is, for instance, a protein. This is at intermediate water levels. So if you start to remove a little bit of water from cells, what happens is these solutes, they're also called compatible solutes, they allow for a preferential hydration layer of water around the protein or the phospholipid, the membranes, thereby keeping their structure. If you now remove the final water, of course, you can't make this hydration shell anymore. And that actually then what happens, and that is below 0.3 grams of water per gram of dry weight. It's kind of a magic number. The hydrophilic molecules are actually the OH groups of the sugars and the LEA proteins replace the water via hydrogen bonding. And thereby they stabilize, so they take the place of water. And this theory or this phenomenon is referred to as the water replacement hypothesis. The second mechanism in which both of them are involved in is the formation of a glassy state. In fact, if you take the seeds and you dry them back slowly and you see what goes on within the cytoplasm, and this we have measured using a small probe, and we look at the rotation of the probe in the cytoplasm, we can see that the viscosity slowly goes up when we dry the seeds up to the point of 0.3 grams of water per gram of dry weight. And then when we dry further, you can see there's a 5 orders of magnitude of the viscosity. The formation of the glass is comparable to the outside of an M&M. It's kind of this very hard structure. Now, what is this glass? It's a solid-like liquid, so it's not a crystal. It has a disordered state of a liquid, but a liquid with extremely high viscosity. And to give you an idea, a normal liquid flows with a speed of 10 meters per second, whereas in a glassy state, it flows 10 to the minus 14 meters per second. So about 30 microns in a century. So it's really something that is really solid. And of course, this means that aging reactions will be severely slowed down. And this is why there is such a high longevity. Now, as I said, we have to also remove potentially damaging molecules. And one of these damaging molecules or one of these damaging organs, in fact, are the chloroplasts. Chlorophyll retention, first of all, is a marker for a truncated maturation program. So if you see green seeds, this often means there might be a problem with your seeds. But yet, if you look at the mutants of SGR, so stay green mutants, it's here in Medicago seeds again, you can see here when we measure the longevity in the wild type seeds or in the SGR mutants, there is no problem. So apparently it's not the chlorophyll per se that is a problem. But yet when we start to knock out genes involved in chlorophyll degradation, we also have a decrease in longevity. And what we have figured out, and this is something that we will, that we hope will come out very soon in Plant Journal, is that we actually, what you have to do is to shut down the photosynthesis during maturation. So you have to deassemble your chloroplasts. That is a prerequisite for the acquisition of longevity. And the gene that actually regulates this is a abscisic acid insensitive 4, which is a transcription factor, an ethylene transcription factor. Then the third part that I talked to you about, the third type of mechanism, is the repair. You have the synthesis of molecular chaperones that during inhibition eliminate aggregated proteins. And one example is here. This is a small Lea protein. This is what we've shown in Medicago as well. What we did is we took the cytoplasm of immature Medicago seeds that have not yet accumulated these late abundant proteins, these Lea proteins. And if we freeze or dry the cytoplasm and we rehydrate, you see these big crystals here. And if you add a little bit of this Lea protein, you see actually that these crystals have mostly disappeared. These aggregates, sorry. So what you can see, it eliminates the aggregated proteins. You also have the activation of DNA damaging, damage signaling molecules that then in their turn activate the synthesis of DNA repair enzymes such as DNA ligases, protein isospartyl methyltransferases, PIMT, and methionine sulfoxide reductases. And this has all been shown for Medicago, for legume seeds. So just as a final slide, in terms of the signaling part, what you have is this ABI4, this abscisic acid 4 transcription factors that is regulating the dismantling of the chloroplast, coordinating the shutdown of photosynthesis and helping the formation of the eoplast. You have via ABI5, another transcription factor, also abscisic acid insensitive, the production of these Lea proteins, the oligosaccharides, that gives you all the protective compounds, and that all leads to the acquisition of this longevity and this seed vigor. And all of this is regulated by another abscisic acid insensitive protein transcription factors, which is ABI3. And just for you to know, if you look in Medicago, what happens with these ABI3 seeds, you can see them here. If you knock out the gene, the seeds are completely green and they're desiccation insensitive. So this is really the regulator that is upstream of this whole seed vigor program. Okay, just a couple of slides on the effect of environment. So what I've shown you, you have the increase in seed quality during physical seed quality during seed maturation. How this is acquired has its effect on how long you can store them, how well they can germinate, how well they can emerge. And all of this is influenced by environment. And in fact, seed vigor is under strong determination of genotypes by environment interactions. They remain poorly understood. We're starting to look at them now. And knowing these genes is important to breed for seeds that are more resilient for climate change. So we really have been working on a lot on it. And the thing that we're particularly interested in is this phenotypic plasticity. Now, what is this? Well, if your seed batch or if your plant is capable of producing different phenotypes according to its surrounding environment, and this is something that is reproducible, we talk about phenotypic plasticity. Just to show you here what it is about, so if you just look at the genetic effect, here you put two different accessions in blue and in red under two different environments. A change in environment can actually give you, for instance, higher longevity or a higher germination. You also can just have a simple environmental effect that you have two genotypes, and irrespective of the environment, one will give a higher effect, a higher longevity, for instance, than the other one. And then the G times E is where under one environment, for instance, you wouldn't see a difference between the seed vigor in both accessions. But then when you put them in a second environment, you actually see that they respond differently. And this is the G times E environment. So as I said, we're quite interested in it. And just some preliminary results that we have that we are writing at the moment using the HubMap collection of Medicago truncatula. We have sown 200 accessions under 4 different environments. And we determined the longevity at 60% relative humidity, 35 degrees. So this is days, so it's an experiment that took a little bit more than a year. And as you can see, We have accessions where the different environments produce seeds with the same longevity, accessions where longevity goes up, or under the same environments where longevity goes down. And getting, of course, doing association studies and getting the genes that are underlying this behavior is something that we're very much interested in. Just one other result that has been published is the work of one of our team leaders, Jérôme Verdier. What he looked at is the speed of germination of seeds that are produced under normal conditions or under non-stress conditions, 20 degrees here in blue, or produced under hot conditions at 26 degrees on average in the greenhouse. And as you can see, the speed of germination, so the time to reach 50% of germination, is differing between the different treatments. When he did GWAS, he found one beautiful QTL here on chromosome 2, and this was underlying a gene that is known as miel, which is a ring type 3 E3 ligase, MIP30 interaction E3 ligase. And what he found is when he put these seeds under control conditions, the mutants of these seeds under control conditions, he found indeed there was a difference in germination speed. And when he put them under hot conditions, he found that there was no difference anymore. So it looks like this is really a gene that is regulating this plasticity of the germination speed. So I come to the end of my presentation, 5 bullet points, kind of take-home message. What I've shown you is that seed vigor traits are important for stand establishment, homogeneous crop establishment, good emergence under adverse conditions, cold or drought, and maintaining viability and fast germination during storage. What I've also shown you is that producing homogeneous and fast germinating seedlots depends on the mother plant. This is really something that is acquired while the seeds are attached on the plant. And this is all related to the activation of the programs that I showed you that lead to their acquisition. The expression of seed vigor is especially evident when you put the seeds under suboptimal conditions. Germinating at 20 degrees or 25 degrees, your seeds, they will all happily germinate. It's when you put them under more difficult conditions, more suboptimal conditions, that you will see there is a problem with seed vigor. Both the growth conditions prior and during seed development and also all the processing treatments after harvesting strongly contribute to the extent of longevity and seed vigor. So this is really important. And I also have shown you that natural genetic diversity needs to be exploited to identify genes involved in seed vigor traits and to understand the effects of environment. Okay, and with that, I would like to thank all the people of whom I showed worked in this presentation, and also all the other collaborators for which I could not show any results, but that we work a lot with. I would like to thank the financial contributors for giving us money to do all this work. And I just would like to draw your attention. There is a book that we published very recently on advances in seed science and technology for a more sustainable crop production, if you're interested in learning a little bit more about seeds. And the final slide I want to show you also as a president of the International Seed Science Society. Next year in Paris, we will have our biennial meeting. Here you see the different subjects, and it will be in the beginning of July. And you're all very welcome to join us. And with that, I would like to thank you for your attention.
Many thanks, Julia. It was a very interesting presentation, and you covered a lot of very interesting topic and a lot of hot topic for what I could guess. Now it's time if there are questions from our guests. Otherwise, I have a couple of them, but let's see if any of our guests has something to ask you. Okay, so I will start with a question for you, and it is you just in the last few slides you talked about phenotype plasticity, and you showed this result about MEL1 locus that could influence plasticity relative to vigor. And this is that's probably because I come from the background of general genomic selection and improvement, and I would like to understand if you think that This is the expected case with very oligogenetic seeds with maybe 1-2 loci that contribute to the effect of seed vigor, or you would expect in general to have many contributing factors?
Okay, yeah, thank you for the question. Well, no, actually, I think the results that I showed you here was really quite surprising. When we do the same study on different seed vigor traits, we see many more loci. And also, depending on the species, we see many more loci. So I think it's more the exception of the rule, and which of course kind of helped to apparently it's a major gene. I don't really know why we did not see the other ones, but it's a major gene involved in this trait. But other traits and other species, we have more loci. But there are two types of genes that we expect to find. One is the genes that are regulating directly the trait and that are temperature sensitive. So, for instance, a gene that produces a protective molecule and that is then producing more or less if temperature is higher or lower. And it can be the regulators. And what we see is we find more the regulators than that we find directly the structural genes that are related to this.
Okay. So you would expect the regulation at least shows more variability, and that's why we are able to intercept those. Okay, makes sense.
Yeah.
Paolo, I see Paolo that raised his hand. So Paolo, you are muted. Okay, so speak up.
Hi, Julia. Thanks for your presentation. We, as a group, we sometimes find difficulties in working with soybean because of bad germination, and I know we are not the only ones. So one thing I wanted to ask you is something I was not really aware of, that is inhibitional damage. So a little bit to know more about that. And secondly, well, I really would like to know a little bit better, for example, people working, in crossing programs. And so how much is important that the harvested seed is dehydrated and kept at very low, let's say at low temperature, let's say below zero. So I wanted to know if for soybean, if we don't do that, the consequence will be much worse than in other species, as I guess.
Okay, yeah, so this has really been studied a lot for soybean, and so there are three factors that are important for this inhibitional injury or inhibitional damage. The first one is about the genotype, because some are much more sensitive than the other. The second one is the initial water content at which you start imbibing your seeds, and the drier they are, the worse it is. And they have this magic 10% moisture content. That's why they say don't dry your seeds, over dry your seeds of soybean, because you might have problems with inhibitional damage. And then the second thing, the third part is the temperature at which you imbibe. You can go very low with your moisture contents of your seed, and if you imbibe at 35 with water with 35 degrees, you have less damage. You don't have much damage. But if you do the same thing, you have very dry seeds and you imbibe them with water at 20 degrees or 15 degrees, that's where you start to see the damage. So there's this interaction between the amount of initial water in the seeds and the temperature at which you imbibe the seeds. And what we know it is linked to, it is the speed of the inhibition. Because all these legume seeds in general, but especially soybean, it increases a lot in volume, especially in the beginning when it imbibes. And this is where you have the problem where your membranes are changing from liquid phase, from gel phase to liquid phase, and you start to have little holes. And if it goes too fast, you actually pull holes in your membranes. And this is where you have your leakage of your solutes. And that is the beginning of the ends, because then some of the cells, the outside cells, are dead. You attract your pathogens because your cytoplasm is running out of the cells, and then if you put them in the soil, this is really where the problems start. And so the role of the seed coat, yes, there is a role of the seed coat, and I'm trying to we don't exactly know. We just know that some genotypes are better than other ones, but we have not. I have not found any papers that really show the link between the hardness of the seed coat or the permeability of the seed coat, except knowing that the speed of imbibition is important, which is of course regulated by the seed coat. I hope that answers your question.
Yes, thanks.
Okay, so let's see. I don't see any other hands up. So I guess that I'm going to stop recording, but not before having thanked again Julia for her very interesting presentation. The presentation will go on our website today or tomorrow, and so everybody will be able to see it again. And I know for sure that I put a couple of notes in, because I would like to see again your what you were talking about, and to deeper a couple of very interesting things you talk about. So I thanks again for your presentation, and thank all the present here and everybody that will watch again our webinars. With everybody else, our next appointment is next month, so let's keep in touch.